Hello student, good morning to one and all. I am Dr. Tariq, Department of Biotechnology, Islamic College, Onembadi. Today we discuss about the microbial technology, practical. So you know very well staining techniques. So a lot of types of that. So one by one we discuss in the class. Actually, the staining techniques, what is the purpose of this stain? The bacteria are microscopic organisms, is it? There are also colorless forms. There are also colorless forms. All the parts, in order to visualize them, to study their structure, shape, and other structural characteristics, it becomes necessary to make them more easily visible. It is one of the main reasons. This means that the structures have been contrasted from their environment so that they can be seen easily. The stain is the dye used to color the living or dead organelles. These are the types of staining techniques. So, there are two major types of the staining technique. Number one is simple staining, that is use of the single stain, and another one is differential staining. Simple staining and a differential staining. The simple staining means for demonstrate morphological shape and arrangement also, but differential staining identification, gram positive, gram staining, like that, gram negative, and acid fast stain, etc. In between, the another one of the special staining techniques are there. This is also the two types. Number one is spore staining and a capsule staining. So these are the basic staining techniques in the microbial species. And another, the acidic in nature. Some of the acidic stainings are acidic in nature. Some of the staining techniques are base, and some of the neutral. Like so, acidic is negatively charged acid. Radicals importance color in eosin, acid fusion, and a malachite green, and a nigrosin, and also the Indian ink. Okay, so these are all the acidic content in the staining compound. Basic content of the staining compound is available. Positively charged basic radicals combines with negatively charged particles in cytoplasm and gives color. So, for example, hematoxylin, methylene blue, crystal violet, and gentian violet. Neutral stains are also available. Both positively or negatively charged imparts different colors to different components. Example, Zimsia stain, Leishman stain, Wright stain. So, all kinds of stains we have in the lab at the same time. We are using these types of staining also utilized in the bacterial identification. Okay. Okay. Next, the staining methods. The staining methods like positively stainings, where the actual cells are themselves colored and appear in a clear background. Positively staining methods. A simple staining, a stain which provides color contrast but gives same color to all bacteria and cells. For example, Loeffler's methylene blue, polychrome methylene blue, diluted carbon fusion. And another one is differential staining. In this stain, which imparts different colors to different bacteria, it's called differential stain, in which contains more than one stains. For example, gram stains, acid for staining, special stainings also. Okay, these are all the methods of positive staining that is called as simple and differential staining methods. The negative stains means where the cells remain cleared, that means uncolored, the background is colored to create a contrast to aid in the better visualization of the image. That is the examples of staining: Indian ink, and another one is nigrosin. These stains are only for background in the stain. The microbes is uncolored, so it is also the visualized the image in the microscopical view. This is called as negative stains. Okay, how can you staining techniques in the number one method applied in the procedure? Smear. The bacterial smear preparation is smear is a distribution of bacterial cells on a microscopic slide for the purpose of viewing them under the microscope. The method is aseptically a small sample of the culture is spread over the slide surface. Number two, this is then allowed to air dry. So you have to apply the slide in the small drops and smear them, then you spread over the slide after the allowed the dry. Then you have to go into third step. The next step is heat fixation to help the cells adhere to the slide surface permanently fixed. That is called heat fixation. And fourth step is the smear is now ready to staining. Smear is now ready to stain. So these are the pictures shows that a drops apply the microbes, then you have to smear the eventually spread out the slide also. These are the one of the method. Okay, next, tissue sections. The sections being embedded in paraffin, it is necessary to remove the paraffin so that a watery stain may penetrate. The paraffin is first removed with xylene. The xylene is then removed with the alcohol, and the alcohol is replaced with water. The staining is then done. Then smear fixation. Heat fixation, we already discussed in the previous slide. Heat fixation, number one is pass air dried smear through a flame 2 or 3 times. Do not overheat the slide. Okay, just go through the 1 or 2 times or 2 to 3 times in the flame. Okay, not for heating the slide. Okay, another is allow the slide to cool before staining. So you have to wait in few seconds. After that you can the cool it down. Okay, the methanol fixation, place air dried smears in a coupling jar with methanol for 1 minute. Alternatively, flood smear with methanol for 1 minute. ஸோ ஏர் ட்ரையர் ஸ்மியர் கம்ப்ளீட் பண்ணதுக்கப்புறம் அந்த ஜார் மூலயமா எத்தனை நாளைக்கு 1 மினிட்ஸ் உள்ளே வச்சு எடுக்கணும். ஆர் நீ என்ன செய்யணுன்னா பிப்பட்டில் எடுத்து ஜஸ்ட்டு க்ளோஸ் பண்ணி சக் பண்ணி எடுத்துகிட்டு மெத்தனால 1 மினிட் அதில் ஃப்ளோட் பண்ணணும். ஃப்ளட்டு ஃபுல்லாக ஸ்ப்ரெட் பண்ணி விட்ருணும். மெத்தனால் எடுத்து அப்ளை பண்ணிடணும். தென் ட்ரைன் த ஸ்லைட்ஸ். அகைன் யூ ஹாவ் டு drain the slide to allow the dry before staining. Then simple staining methods. This is the first steps in the simple staining method. Methylene blue. First methylene blue. It's one of the dye. It is generally the most useful. It shows the characteristics morphology of polymorphs, lymphocytes, and other cells more clearly than to stronger stains such as the gram stain or dilute carbol fusin. Next stain is methylene blue, that is called polychrome methylene blue. This is made by allowing low-first methylene blue to ripen slowly. The slow oxidation of the methylene blue forms a violet compound that gives the stain its polychrome properties. Then the ripening takes 12 months or more to complete, or it may be ripened quickly by the addition of 1% of potassium carbonate K2CO3 to the stain. It is also employed in McFadden's reactions. Then, in contrast to the blue staining of most structures by the methylene blue, the violet component stains acidic cell structures red purple. Example, the acid capsular materials of the anthrax bacillus in the McFadden reaction. The dilution carbol fusion method is made by diluting Zilson stain with 10 to 20 minutes its volume of water. The stain for 10 to 25 seconds, then wash. It will wash the water with the water. Overstaining must be avoided as this is an intense stain and a prolonged application colors the cell protoplasm in addition to nuclei and bacteria also. These are the fusion method, carboxyl fusion method. Then what do you want in the staining techniques in requirement? The methylene blue and carbon fusion, distilled water, and microscope, wood oil, fixed smear. So these are the essential for your requirements in the staining techniques. The procedure is make a thin smear on a slide. First step, heat fixes the smear by passing the slide 2 to 3 times gently over the Bunsen flame with the smear side up. Then pour methylene blue over the smear and allow it to stand for 3 minutes. Then you have to go in, wash the stain, smear with the water and air dry it. Then you are going to observe the smear first under the low power of 10 power x objective in the microscope. Then under oil immersion 100% 100x objective lens. Okay, then you observe the presence of organisms and also the cellular content in the sample given the microbial samples. The very simple methods. First of all, இன்னொரு தடவை சொல்கிறேன் கேட்டுக்கோங்க. ஃபஸ்ட் எடுத்தோன்னா ஒரு ட்ராப்ஸ் வந்து மைக்ரோபியல் சொல்யூஷன் வந்து ஒரு ட்ராப்ஸ் வச்சுக்கோங்க. அதுக்கப்புறம் என்ன செய்யணும், அதை ஸ்ப்ரெட் பண்ணி விடுங்க. ஸ்ப்ரெட் பண்ணி விட்டு ஹீட் ஃபிக்ஸ் பண்ணிடுங்க. புன்சன் பர்னரில் ஹீட் ஃபிக்ஸ் பண்ணிவிட்டு லைட்டாக கூல் பண்ணதுக்கப்புறம் மெத்திலின் ப்ளூ வந்து அது மேலே ஸ்ப்ரெட் பண்ணி விட்ருங்க. அதாவது அது ஃபுல்லாக ஓவர்ஃப்ளோ பண்ணி விட்ருங்க. அது ஒரு 3 நிமிஷம் வச்சிங்கன்னா ட்ரை ஆயிரும். அதுக்கப்புறம் ஏர் ட்ரையில் கூட பண்ணலாம், ஃபேன்லேயோ இல்லாட்டி ஏர் ட்ரையர் வச்சு யூஸ் பண்ணிக்கலாம். வச்சு முடித்ததுக்கப்புறம் அதை என்ன செய்யணுன்னா டைரெக்டாக வாட்டரில் வாஷ் பண்ணிக்கலாம். இல்லைன்னா அப்படியே கொண்டு வந்து நீங்கள் என்ன செய்யலாம், வாட்டரில் வாஷ் பண்ணிக்கணும். ஸ்டெயினிங் தான் ஸ்மியர் பண்ணது எல்லாத்தையுமே வாஷ் பண்ணிவிட்டு ட்ரை பண்ணணும். ட்ரை பண்ணதுக்கப்புறம் மைக்ரோஸ்கோப்பில் வச்சு அதை பார்க்கணும். ஃபஸ்ட்டு 10x வச்சு பார்க்கணும். அதுக்கப்புறம் வந்து 100x, 40x இருந்தால் 40xயில் வச்சு பார்க்கணும். அப்புறம் 100xயில் வச்சு பார்க்கணும். இதை வச்சு பார்த்தீங்கன்னா நீங்கள் வந்து கிளியராக வந்து அந்த செல்லுலார் கண்டென்ட் வந்து சாம்பிள்ஸ் வந்து நம்ம பார்க்கலாம். திஸ் இஸ் த ஒன் ஆஃப் த easiest method, that is called simple staining method. So the picture shows that the clear cut of the staining in the methylene blue color. These are the basic steps in the 100x. This is the picture shows the 100x method. Okay, thanks to watching. Thanks.